Hi, everyone. Let's get started. This is my co-host, Assistant Professor Amanpreet Batwa from the University of Montreal. And I'm Thomas Yeo from National University of Singapore. It is my honor to introduce our next keynote speaker, Associate Professor Anastasia Yendiki. Anastasia is the Director of Circuit Analysis at the Martino Center for Biomedical Imaging at Harvard Medical School. In the past 10 years, she has made many important contributions to diffusion tractography. For example, Anastasia developed the Tracular package, which is a toolbox for diffusion MRI preprocessing, tractography, and statistical analysis. Her package has been widely used to study development, aging, and a wide range of mental disorders. She has also done very important work validating diffusion tractography against anatomical tracing in non-human primates and optical imaging in human brain samples. So today she will tell us about her vision for diffusion tractography. Please welcome Professor Anastasia Yendiki.
Thank you, Thomas, for the kind introduction. And a big thank you to the program committee for their trust in me. Ever since I started coming to OHBM, it's always been a dream of mine to one day be the opening act for Club Night. And here we are. It was a risky choice on the part of the organizers. So this talk is going to be an amalgam of lessons learned, both from developing tools for in vivo tractography studies and from comparing tractography to anatomy in ex vivo studies. Tractography came about from two fundamental ideas. First, that you could use MR signals sensitized to the diffusion of water molecules in different orientations to detect the preferential orientation of diffusion in every voxel in the brain. And second, that you could follow those orientation vectors from voxel to voxel to connect the dots in the brain and reconstruct images of fiber pathways. The early excitement about tractography came from the first studies that showed that this technique was able to reconstruct known white matter pathways, like the fornix and the thalamic tracts shown here, for the first time from an in vivo MRI scan. Until then, these pathways could only be identified post-mortem, for example, with anatomic tracing in non-human primates or by physically dissecting human post-mortem brains. And early comparisons of in vivo tractography to these post-mortem techniques showed that, for the most part, tractography could get the core part of the large highways of the brain correctly, but that to get to that part correctly, we had to do quite a bit of manual editing to remove the parts that tractography got incorrectly. And so this set off an effort to come up with standardized protocols for doing this manual editing. These were called multi-ROI protocols because they define each major white matter pathway on the basis of 2 to 3 ROIs that the pathway goes through and maybe a couple of ROIs that it doesn't go through. And these multi-ROI protocols are still the basis for the majority of automated methods for reconstructing tracts of interest today. The automated flavor of the multi-ROI approach simply takes the ROIs from an atlas and maps them to the individual instead of having to draw them manually in the individual. So let's see how that works. The diffusion orientations from the subject's own diffusion scan are used to seed tractography either everywhere or in part of the brain. And this step is agnostic typically to what actual tracts we are going to reconstruct. In a second step, some ROIs are mapped from a template brain to the individual, and only the streamlines that go through these ROIs are retained. If everything went well, and the original tractogram from the individual subject did contain some streamlines that go through these ROIs, then we're good to go. We have our tract of interest. But if things didn't go so well, and this depends both on the quality of the individual data as well as the degree of difficulty of the specific bundle, then we might not have any streamlines in the original tractogram that go through these ROIs, and then there's nothing we can do. We introduced a different approach for automated tract of interest reconstruction that is not a multi-ROI method. It's called Tracula, or tracts constrained by underlying anatomy. It models each tract of interest as a curve with a small number of control points. And it tries to fit the shape of this curve to two different sources of information. One source of information is the diffusion orientations from the subject's diffusion data. So Tracula tries to come up with curves that are well aligned with those orientation vectors. Another source of information is an anatomical segmentation of the same subject from the subject's T1 data, typically from using FreeSurfer on the T1 data. Tracula tries to come up with curves that are not only well aligned with the diffusion orientations, but also fit our prior knowledge on which labels from the anatomical segmentation are neighbors of this tract of interest in different directions, to the left, right, anterior, posterior, etc. So Tracula knows in advance what tract it's going to reconstruct, and it uses information about the anatomy of that tract in the tractography step itself. This information comes from a set of training subjects where the same tract has been labeled manually, and that come with their own FreeSurfer segmentation, so those anatomical priors are computed from those subjects. The control points of the curve are perturbed to produce many curves, and then the curves that fit both the diffusion orientations and the anatomical priors at once are retained. The first version of Tracula, which was included in FreeSurfer 5.1, could reconstruct 18 tracts that covered only 18% of white matter. The most recent version, that was included in FreeSurfer 7.2, can reconstruct 42 tracts that cover 82% of white matter. It is described in this recent paper by Chiara Maffei et al. In our favorite journal, Neuroimage. The better coverage comes not only from having more tracts, but also from having fuller and more detailed model of those tracts. In the old version, we had training data that came from annotating the tracts manually in conventional scans of that era. And this is an example of what the SLF, the entire SLF, looked like that we could get in one of those old scans. These days, we are using manually annotated examples of the tracts from scans that were acquired on the Connectome scanner at MGH. And this is an example of what the SLF 1, 2, and 3 look like in one of those manually annotated subjects. Our goal here was to use these high-end data to obtain the training data for Tracula, so that Tracula can then reconstruct the same tracts accurately in lower-end data that can be acquired in regular scanners. And as described in detail in Chiara's paper, what we did to show that was we annotated the tracts manually using the complete MGH HCP data sets with Bmax of 10,000 and 512 volumes, and trained Tracula with those manually annotated examples, and then reconstructed the tracts automatically using only the lowest shell, the B equals 1,000, 64 direction, or even just 32 direction data from the same subjects. The good news is that with both the Tracula and the multi-ROI approach, it is possible to reach very high sensitivity, up to 89%, using these lower-end data when you compare the tracts to the manually annotated example from the high-end data. However, to achieve that high sensitivity, each reconstruction method has to incur a different level of reconstruction error, which we quantify as the Hausdorff distance between the automatically reconstructed and manually labeled tracts in the same subject. For Tracula, this reconstruction error to achieve the high sensitivity of 89% is a couple of voxels, 3.5 millimeters. And it gets about 20% higher when we use only 32 direction data. With the multi-ROI method, this reconstruction error to achieve the 89% sensitivity is somewhere between 2 and 3 times higher, depending on whether we use 62 or 32 direction data. And the reason for that is that the multi-ROI method has to run the tractography on the individual data in an agnostic manner, so it really relies on the quality of the individual data. Whereas Tracula uses the rich information that it gets from the training data from the Connectome scanner to make a more informed reconstruction of the same tracts in the individual subject's data. For now, we have focused mainly on the big highways of the brain. But for our next step, we want to move on from the big highways and into the deep, dark alleys of the brain. And this is work, again, by Chiara in collaboration with Fuyishui Wang and Suzanne Haber on the anatomy guidance. So in this case, Chiara wanted to dissect a set of small, deep brain bundles that are important for psychiatric applications. And first, she tried reconstructing them using the MGH HCP data with a B equals 10,000, which, however, are collected at a 1.5 millimeter resolution. And it turns out that that resolution is not high enough for some of these deep brain bundles. The big bundle shown here, the big round one, is the fornix, and it's shown for reference because it's one that's big enough to reconstruct at both levels of resolution here. So on the left, we see the 1.5 millimeter resolution Human Connectome Project data, and on the right, we see a data set that was collected by Fuyishui on the Connectome scanner with a G-slider sequence at resolution of 0.76 millimeter isotropic. So the smaller bundles, like the stria terminalis, the fasciculus retroflexus, the mammulotegmental and mammulothalamic tracts, can only be reconstructed at the sub-millimeter resolution level. And this shows the importance that spatial resolution is going to play as we go from the highways to the smaller bundles. Unfortunately, the data set on the right was acquired by scanning a single subject for 15 hours total over multiple sessions. Not exactly an everyday practical acquisition. But going forward, we would like to take advantage of a combination of super resolution methods as well as the anatomic priors that I showed you earlier, so that one day we can use the high-end training data that we get from manually annotating these bundles in the sub-millimeter resolution data to be able to reconstruct these tracks more reliably in scans that we can acquire in less amount of time. So far, we have talked about supervised tractography, where we know what tracks we want to reconstruct, and we train an algorithm to do that. If this was structural MRI, we would be done. We could declare victory and go straight to club night. Because that's what we do when we segment MRI data. We know that there's a thalamus, an amygdala, et cetera, et cetera. What would it be like to use a structural segmentation algorithm in unsupervised mode? So we don't know what structures are there in the gray matter. The algorithm segments the gray matter. It gives us a new nucleus that has never been documented before. We give it a new name, the nucleus of Glasgow, and we publish it. We typically wouldn't do that with structural data, but we do that sometimes with diffusion data. And that's when we run tractography in unsupervised mode. I don't want to imply here that this is a hopeless task. I will actually spend the rest of my time trying to find clues for how to do that. But I do want to point out that it's a very difficult problem, and so it's no surprise that we haven't solved it yet. And I also want to point out that these are two different tasks, the supervised and the unsupervised task. So even if we can't fully trust tractography in the unsupervised task, that doesn't mean that we can't trust it for the supervised task. There we have good tools that can do that well and can do it well with not particularly sophisticated acquisitions. So to get clues for how to fix tractography, we need to figure out where it fails. And of all the invasive techniques, the one that does exactly what tractography aspires to do non-invasively is anatomic tracing, where a tracer is injected in the cortex and it propagates down the axons. We have been collaborating with Suzanne Haber, whose group has been doing these invasive tracer injections for quite a while, to collect high-resolution diffusion MRI scans on some of the brains that have received the tracer injections. We scan these brains ex vivo at the Martino Center, send them back to Suzanne, and then she sections and stains the brains to visualize the tracers. This allows us a point-by-point comparison of tracing and tractography in the same brain. Obviously, this isn't applicable to the human brain, so we can't do a point-by-point comparison with tracer there, but we can take advantage of topographies that Suzanne's group has been mapping carefully with tracer studies over the past few years, and we can evaluate the extent to which tractography can replicate the topographic organization of smaller fiber bundles in the white matter based on their cortical area of origin, how well it can replicate those topographies that are found from the tracer studies, and then translate that information to see if we can get the same topographies in the human data. So there is potential for translating these kinds of studies to validate tractography even in the human. Several studies have compared anatomic tracing to diffusion tractography in the past, and they're arranged here in terms of the b-value and the spatial resolution of the diffusion data that they used, and the size of the marker is the angular resolution, the number of directions. Most of these studies have compared only the terminals to the endpoints of tractography, and not the full trajectory of the axons. And those are the empty markers. And that's because most of the studies, and those are the gray markers, have compared anatomic tracing and diffusion MRI from different brains. So typically there's a database of tracer injections, and that database only contains information about the terminals, so they can only, those studies can only validate the endpoints of tractography. This is very useful for determining if tractography has gone wrong, but when it has gone wrong, we don't know where along the way it went wrong. And in the data that we have been collecting with Suzanne, we have anatomic tracing and diffusion MRI in the same subject. And in contrast to the few studies that had done that before, we are not focusing on M1, but we're focusing on injections in frontal cortex that are quite challenging for diffusion tractography. What are some of our key findings? One finding that we find pretty consistently is from the comparison of deterministic versus probabilistic tractography. When you compare them at the same false positive rate, probabilistic tractography has a higher true positive rate in general than deterministic tractography. And we find this consistently regardless of the Q-space sampling scheme and the orientation reconstruction method. There is some converging evidence for that from other studies that have compared the endpoints of tractography only. We have used these data with the tracing and diffusion in the same brains for the Iron Track Challenge with the participation of these wonderful individuals. I would like to thank them because we made them do quite a bit of work over 2 rounds of the challenge to really narrow down our findings and disentangle the effects of different aspects of the analytic pipeline on the accuracy of tractography with respect to the tracer. And I also want to thank Umenta for hosting the challenge on their cloud-based platform. The teams were asked to fine-tune their tractography pipelines to maximize accuracy with respect to anatomic tracing in an injection in the frontal pole, which we call the training case. And then they applied that optimized pipeline to an injection area in the VLPFC that we refer to as the validation case. This way, we tested both the accuracy of the different pipelines and their robustness with respect to different seed areas. So we came up with some recommended practices for analyzing multi-shell diffusion MRI data. One finding consistent with those previous ones was that for the most part, probabilistic methods outperform deterministic ones. And by the way, you can follow this QR code for complete rankings. As far as orientation reconstruction methods, the Roomba spherical deconvolution method was what was used in the winning pipeline. And perhaps surprisingly, we found that a simple post-processing step of filtering the tractogram with a 3D Gaussian filter improved the anatomic accuracy across most pipelines. Again, this has been published in Euroimage in the last few weeks by Chiara. So another key finding has to do with the failure modes of tractography. What are the areas and what are the fiber configurations where different analytic pipelines fail consistently? And I'm going to show you a video that Chiara made to highlight some of these trouble spots. So what you'll see here is in red, there's a tractogram from, let me restart that. So red is a tractogram from one of the teams. Yellow is the true positives, the voxels that were visited both by the tractogram and by the tracing. And blue is the false negatives, so areas that were visited by the tracer injection but not by the tractography. And you'll notice that there will be places where the blue, the tracer, and the red, the tractogram, diverge. And this will happen consistently in areas where there are branching fibers, where the fibers from the injection site branch to reach different destinations. The first one is going to be very close to this injection site in the frontal pole. It's going to be the short-range fibers that fan off of the main trunk of the injection to reach nearby areas of frontal cortex, and tractography fails to find these. And then we'll see other branch points. So again, the blue is the tracer. Those are those short-range fibers that tractography cannot get. Then there's going to be a branch point between the cingulum bundle and the corpus callosum, where tractography gets only the latter. Then a branch point between the internal capsule and the external capsule. Tractography gets only the former. And this sharp turn in the uncinate. And here comes the VLPFC injection. We'll see very similar patterns, that cingulum and corpus callosum branching. And then another branch point with thalamic versus brainstem fibers. So usually tractography follows only one of these paths and not the other. And here are those trouble spots. So it's important to note that crossing fibers here will exist everywhere along these fiber bundles. But the consistent errors across different analytic pipelines don't occur just anywhere. They systematically occur in these areas where the fiber configurations actually diverge from crossings. Where does this come from? And is it something that will ever be solved because our data, diffusion data, will improve? For the past 20 years, the tractography community has been focused on solving this crossing fiber problem. This is done typically by fitting an orientation distribution function that expresses the probability of a fiber crossing a certain voxel at a certain angle. As our data have improved in terms of SNR and angular resolution, these ODFs have gotten more and more accurate. However, an accurate ODF is not enough. It's enough for a crossing, but not for every other configuration. There are multiple configurations of two different fiber bundles in a voxel that give rise to the same ODF. The ODF tells us in all these cases there's some diffusion like this and some diffusion like that. When the tractography algorithm reaches this voxel, it doesn't know which of the available ODF peaks to follow, so it has to guess. Typically, it goes as straight as possible, and that will give us the correct answer if the correct answer is a crossing. It will not give us the correct answer if the correct answer is fanning or turning or branching. This doesn't have to do with the quality of the data. Even if we had a noiseless continuous space ODF, so even if we had infinite SNR and infinitesimal angular resolution, we would still have this ambiguity with the ODF. So we have to think a bit beyond the ODF, either find another way to choose between these peaks, or find another way to generalize the ODF, or find something entirely different. And this is where the tracer data offers important insights for what fibers do that actually diverge from these cartoon images of fibers that we're used to looking at. One is the branching, and I'm being a little cheeky here, it's not new. In fact, if you read a description of a fiber bundle in any anatomy paper, it's essentially described in terms of these branch points. Because each gray area is connected to multiple other gray areas, gray matter area, we know that there's going to be branching because there's one to many. And in fact, in the tracer data, we see these branch points often, and we see them clearly. So it's, as we saw, it's these areas where tractography errs consistently, and so it's something worth looking into more in the future. Another important point that we see once we start looking at fibers under the microscope is that not all bundles are all that bundled. So we tend to think of bundles as a bunch of fibers that start from the same start point, stick together, and reach the same destination. This is very much the case if we look in the internal capsule. There, the fibers from the injection site stick together. Also, more or less true in the corpus callosum. But if we look at other pathways, like the uncinate fasciculus, we see that in between the fibers from the injection site, there are big gaps. And in those gaps, there are fibers running from other cortical areas. So this has potential implications for methods that try to disentangle different bundles based on their microstructure. Because here we'll have multiple different bundles in the same fiber population of an ODF. So this is something to further look into in the future. Another way in which real fibers are different from the cartoon pictures or tractography pictures that we look at has to do with superficial white matter fibers. Usually we call those U fibers. The U-shaped ones are the ones that go from gyrus to gyrus, and they're easier to get with tractography. But if you look at real fibers, there's all kinds. So here we have an injection site in a gyrus with the fibers, short-range fibers terminating in a sulcus, so a C shape. Here we have fibers from somewhere between a gyrus and a sulcus traveling towards a gyrus, so more of a sigma shape. And you'll see that here they travel inside the gray matter. Here they travel just underneath the gray matter and the superficial white matter. So it's important to know when we clean up our tractography data too much and keep only the pretty U-shaped things, that we may be cleaning up true anatomy. So one important next step as we look at all these things, one question is, well, how often does this happen and how often does that happen in the brain? So it's important to come up with automated methods for labeling the tracer data, because that is currently our bottleneck. It takes a long time to do the manual annotation. And Vanathy from our group has been working hard on Suzanne's data to train machine learning models to do that. So these are some very preliminary results where she has trained a dual task network to both segment the outlines of the fiber bundles and segment the fibers from the background within the bundles, which is important because, as we saw, in some bundles, the fibers will be very close to each other, and in others, there'll be gaps. So this is a first step towards being able to quantify these things, as well as to accelerate the pace and the number of different cases that we can use for validation studies. With anatomic tracing, we get all the axons from a specific injection site along their entire trajectory. But to model the diffusion signal in a certain voxel, we need all the fibers that come into that voxel, regardless of where they came from. And we cannot get that from tracing. And it's also helpful to be able to do this sort of quantification in the human brain directly. For this, we use a technique that's called polarization-sensitive optical coherence tomography, or PSOCT, which at the Martino Center has been spearheaded by Hui Wang. It looked like this a few years ago. Now it looks much more civilized, the new system. PSOCT uses polarization of light to probe birefringence, a property of anisotropic tissues. It's the difference in refractive index, parallel versus perpendicular to the axis of anisotropy, which is the axis of the myelin sheaths. So in brain, this birefringence is due to the regular pattern of arrays of lipids and proteins in myelin. What we get from PSOCT is direct measurements at the microscopic resolution of the optic axis orientation, these color maps, and a bunch of other contrasts like retardance, which is a measure of the myelin content. There are two key strengths to the PSOCT approach. One is that it does not require registration across sections. In many other approaches, like histologic myelin stains or PLI, you have to first section the tissue and then image it. Here, we image the superficial layer of the tissue, the block face, first, and then cut it. So the slices come out aligned right out of the scan. Another key benefit is that, unlike histologic myelin stains, you do not need to do any image processing to segment the axons and infer their orientation. So you get this vector map of axonal orientations out of the scan. So right out of the box, you can run tractography at the microscale and reconstruct fibers and follow them across the brain. And the technology has been evolving over the last few years. One limitation was that with PSOCT, you wouldn't get any optic axis contrast in the cortex. Thankfully, with this new technique developed by Chao Liu, who's a postdoc working with Hui at the Martino Center, that involves submerging the tissue in refractive index matching solution prior to imaging, there is now much better contrast, both in gray and in white matter, and allows us to follow those fiber orientations as they enter the cortex. Another key limitation up to now was that we could only get in-plane orientation measurements. So the color maps that you saw are the orientation of the fibers in the plane perpendicular to the angle of incidence of the light. This can be fixed by having 2 light incidence angles and collect twice as much data or 3 times as much. And these preliminary results from Chao are very encouraging, and we're very excited about this new development. He imaged a sample of the corpus callosum, tilted either by 0 or by 30 or minus 30 degrees, and imaged it with multiple incidence angles, and was able to show, first of all, that the in-plane orientation is consistent, and that the through-plane orientation is accurate compared to how the tissue is actually tilted. This is going to be a very important step towards being able to follow axonal orientations in 3D across the brain. At the moment, we can only apply PSOCT to small fields of view, small samples. So we honed in on one of those problem areas where a bunch of fibers branch and cross, following up on the findings of the tracer studies. This is a branch point between the internal and the external capsule. We collected 9.4 Tesla, we collected diffusion data in a 9.4 Tesla preclinical scanner in a way that allows us to compare different Q-space sampling methods, multi-shell, single-shell Cartesian. And also we downsampled the data from their original 0.25 millimeter resolution to 0.5 and 1 millimeter and 2 millimeters. And we acquired PSOCT data from the most complex area of the sample. What we found was that the biggest effect on the accuracy of the diffusion orientations with respect to the optical orientation measurements was actually the spatial resolution. And the biggest jump happened when we went from 2 millimeters to 1 millimeter. The next bigger effect was the orientation reconstruction method that we used to obtain the diffusion orientations. And the very last one was which of these Q-space sampling schemes we use, whether it was single shell, multi-shell, et cetera. This is work by Robert Jones, again published in Neuroimage, a little predictable here. So then we looked at different diffusion voxels with different fiber configurations, crossing, branching, and coherent fibers, to see where those effects came from. The jump in accuracy that we see by going from 2 millimeter to 1 millimeter resolution came from the branching areas. There, all the different orientation reconstruction methods, which are shown with different colors, do about the same. They play a role, the orientation reconstruction method does play a role in the crossing area, predictably, because that's what they're designed to do. But for the branching and as a sanity check also for the coherent area, they all do the same. Going forward, it will be important to also use these data as another way to quantify the different fiber configurations that we see in the brain and how often we get these non-crossing patterns that we have to pay attention to. So the simplest approach would be to just take the orientations from the optical data at the microscopic resolution and just compute fiber orientation distributions, or FODs, and try to learn how to infer the ground truth FODs from the diffusion data. However, then we end up with FODs, which is a crossing fiber model, and we'll have the same issues. So we've been trying to think about other ways to capture the different fiber configurations in a voxel that go beyond the ODF. One thing that we tried a few years ago was to run tractography on the microscopic orientation vectors from the optical imaging and to quantify the amount of fibers that connect each pair of edges of the diffusion voxel. And we found that this was a parsimonious enough representation with just a few parameters that, however, captured the anatomical detail of this sample. So going forward, this is something that we will be pursuing further, trying to figure out good models that go beyond the crossing fiber. To conclude, I've went over a lot of things. Supervised versus unsupervised tractography, these are two very different problems, as we saw. For the supervised problem, we have good solutions. For the unsupervised problem, we still need some work to do. And to make progress in unsupervised tractography, we need to go beyond the crossing. This is a consistent message from all our validation studies, as well as also coming from other studies by other groups. And to exploit advances in learning, to take advantage of all these new algorithms, we need ground truth that these algorithms need to learn from. And ex vivo imaging is the perfect source for that. From anatomic tracing in non-human primates, we get key insights for how to best harness these newer modern microscopy techniques like PSOCT that I showed here and others. And our ultimate goal going forward is to use the ex vivo data to inform and improve our methods for in vivo tractography. And here I'd like to take a moment to thank the people whose work I showed today. So Tiara, Robert, Vanathi, Zhao Hui, and of course, Suzanne. This picture looks like we're about to rob a brain bank, but actually it's not. It was our first launch in Harvard Yard after the lockdown, so in summer of 2020. It's not as intense as it looks. And I will end with some resources. If you want to find out more about trachea, there's tutorials and documentation on the FreeSurfer Wiki. And last but not least, my new hobby. If you're interested in a gentle introduction to diffusion MRI, you'll find this interactive notebook-based tutorial that uses freesurfer.jl, a brand new package written in the Julia language. Thank you very much for your attention, and I look forward to your questions.
Thank you, Anastasia, for that wonderful, very clear talk. There are several questions, and I hope we can get through several of them before our time's up. So a couple of questions along the same vein is, can trachea handle subjects who are very different, like if they have a tumor or some form of neurodegeneration?
Yeah, tumors are always a big issue with supervised methods. If it's neurodegeneration, that is less of a problem because trachea doesn't constrain the tracts to have the same size or shape or integrity in the test subjects as in the training subjects. It only uses information from the training subjects about the relative position of the tracts with respect to the anatomical structures. So unless the tract has rerouted itself through a completely different part of anatomy, which might happen with tumors, but unlikely to have happened with simple neurodegeneration, then it should not be a problem.
Thank you.
So the next question is, someone's saying there's so many tracking softwares. How do we decide which to use? Should papers show their results replicate over more than 2 pipelines?
Yeah, I mean, it's always good to show more results, but it's also a big workload for authors and for reviewers. That was the logic behind the Iron Track Challenge, and we hope to get the resources to build it into a platform that would be plug and play for anyone who develops a new method to be able to test it out on these data. Obviously, we need to collect additional data because nowadays people don't use just a simple diffusion scan. They also use microstructural scans that have different types of encoding. So we need to collect a more complete data set with some new monkeys, and we're in the process of doing that. So we hope to have a tool available.
Just to follow up, so in your Iron Track Challenge, so in the end, what was the top package? And would you just recommend that everyone should just use it? Or, or the packages are close enough that it is hard to tell?
So it was, nobody uses a single package.
Okay.
So they put together things from different packages. And so we have a set of recommendations. The main one was probabilistic methods, regardless of the orientation reconstruction methods and other details tend to outperform deterministic methods. So that's something that seems pretty clear. In terms of the different spherical deconvolution variants, etc., that seems to make less of a difference. One thing that was important is that we see that the methods that give the sharpest ODFs are not necessarily the most anatomically accurate methods. And that's interesting because usually the sharpness of the ODF is used as a metric of quality of the reconstruction method. But those fanning and turning, et cetera, areas are easier to capture if you have an ODF that's not super sharp, and if you use probabilistic tractography to sample more directions.
Okay, so this question is quite interesting. Is it known whether crossing fibers can exchange information?
It is very interesting. If you look at tracer injection studies as an example, the tracer doesn't jump from one fiber bundle to a crossing fiber bundle. It stays within the axon. So it's not obvious to me how the exchange of information would happen, but yeah, I'll always leave that possibility open, I guess.
Thanks. How is Tracula different than, here's a question from the audience. How is Tracula different than using exclusion or white matter mass in FSL?
Yeah, so I tried to explain that earlier, but I went too fast. So it doesn't use ROIs to constrain the tracts. With the inclusion-exclusion masks, you run the tractography and you apply those ROIs to the tractography, which is run agnostically. With Tracula, the tractography step itself has information about the tracts that you want to reconstruct because it's a Bayesian framework that includes a likelihood term that tries to fit the shape of the tract to the diffusion orientations and a prior term that tries to fit the shape of the tract to its anatomical neighborhood the way it has learned it from the training subjects. That's like the 15-second explanation.
So the next question from the audience is, can Tracula be used to study longitudinal changes in connectivity?
Yeah, so we have a longitudinal stream. So now it depends on what you call connectivity, because if you want a connectivity matrix, that's the unsupervised flavor of tractography. And Tracula doesn't do that. Tracula does the supervised approach where you have specific tracts of interest, and yes, you can follow the microstructure of those tracts of interest longitudinally. We have a dedicated stream for doing that, which instead of reconstructing the tracts in each scan from each time point independently as though it were a cross-sectional data set, it actually reconstructs the tracts jointly, taking into account all the data from all the time points of the specific subject at once, and we've shown that this improves both test-retest reliability and sensitivity to disease effects.
I'm going to not follow the rank exactly. I have a question is curious, which is that you're using, for validation, you're using both OCT and like the tracing in maquettes.
Yeah.
Is there, what are advantages and disadvantages of both?
Yeah,
like,
so yeah, that's a great question. So in general, it's important to note that there isn't any single ground truth approach to getting the ground truth that gives you the entire ground truth. So the tracer is very good for giving you all the projections from a single gray matter area to all the areas that that area projects. But if you look in a voxel, you'll only see the fibers that come from that injection site. You won't see the other fibers that are crossing or branching or whatever. No, you will see the branching because they all came from the same cortical area. But if you wanted, let's say, so if you want to validate tractography, streamline tractography, it's perfect because it does exactly the same thing. If you wanted to validate orientation distribution functions, where you want all the fiber orientations in a voxel, that's where something like PSOCT is more suitable. Does that make sense?
Thanks.
So there was a question about how does the amount of tracer injected affect what we see? And I'll throw in my question, do we have a quality control on the degradation of the brain?
Yeah, so
before-
my quality control is called Suzanne Haber. So this is not something that you should try at home. It's something that requires a lot of expertise and a lot of years of experience and doing it again and again and again. And yes, there are things that can go wrong. So sometimes you have good transport and sometimes you don't. And so somebody with the expertise to be able to recognize that will be able to tell you like, oh, this didn't go anywhere, so we won't use that, and this did. Yeah.
What's the test retest for Suzanne Haber? I'm just kidding.
She's pretty consistent.
I think it's actually 5 o'clock, so let's thank the speaker again.
Thank you.
Thank you all for coming.
Thank you.